Good morning, everybody. I'm Nicola Bonzanni, and I work at the Netherlands Cancer Institute and at the Vrije Universiteit in Amsterdam. And this morning, I will tell you something about a process called hematopoiesis. So the word hematopoiesis comes from ancient Greek and literally means making blood. So blood, as you might know, is not liquid, but is a fluid mixture of compounds and cells, actually many different cell types. And one thing that I found amazing about the hematopoietic process is that all these different types of blood cell types, they all differentiate from a single hematopoietic stem cell, or HSC. And as you can imagine, the differentiation process that leads from this single cell to all kind of mature blood cell types is a quite complex one. And it's very tightly controlled, and sometimes it might fail. And when it fails, then you get an array of diseases. For instance, you can get anemia when you cannot properly differentiate red blood cell types, or you can get leukemia when you don't manage to mature your white blood cells. So understanding how this differentiation process works is a stepping stone to cure these diseases. But so what actually we do know about differentiation, the hematopoiesis process? So we know that there are 11 transcription factors which are intimately involved in the differentiation process. And there are 39 regulatory interactions between those transcription factors. 39 regulatory interactions which are being experimentally validated, which means that they came from ChIP-seq, and subsequently those were validated in vivo in mouse embryo to check whether those regulatory regions were active specifically during hematopoiesis. And on top of that, we have also quite a few gene expression profiles for mature cell types and also intermediate cell types. So given this knowledge, we had two questions. Would it be possible to build a model based on this knowledge which can recapitulate, at least partially, the differentiation process? So that was the first question. And the second question is, could it be possible then to use this model to then make predictions and increase the current state of knowledge about the differentiation process? So in order to tackle these two questions, we started a collaboration in between the ENFAN project with the Bertolt Gottgens group in Cambridge, UK. And when we asked them to tell us something about these 39 regulatory interactions, they provide us a graph, directed graph, which looks something like this. And as you might, when you look at it, you can think that's a pretty intuitive graph. When nodes are genes and interactions are arrows, then you can imagine that A has a positive interaction on C and so on. But it's also very ambiguous because we didn't have any clue about the dependencies between A and B, for instance. Are those both necessary to promote C or not? So we decided to shift to another representation, the one on the right. And by doing so, we introduced a new node here. And now the claim is that this new representation is now intuitive, explicit, and even executable. And this representation is based on Petri nets. So in very brief, what Petri nets are a bipartite graph in which you have two kinds of nodes. You have the rounded places here, which represent the resources of your system. In our case, those would be genes. And the transition, the square boxes, which represent the active parts of your system, so the interactions. And by using those, we can now still represent promotion and inhibitions as we did before, so we keep the same level of intuitiveness. But now we can also do something like this. So now we can explicitly, even graphically, represent independent promotions, like in this case, where A, for instance, can by itself promote the expression of B, or we can represent the difference, and so when A and C are both needed actually to promote the expression of B. So what we did was fly into Cambridge, then talk with a member of Bertolt Gottgens group, and explain, like I just explained to you, this very simple concept. And after a couple of hours, what we come up with was something like this on the whiteboard. So they were able to understand this formalism and encode all the knowledge that has been gained in this field in their lab and in other labs on this whiteboard. And the interesting thing is that this is also a formal language, so that allows us at the same time to translate what they were drawing on the whiteboard actually on the computer, on this sort of network. So we ended up with this network in our computer, but that's just a static representation of the knowledge. So the question is, can we actually derive some dynamical properties? Can we actually execute it? So in order to do so, we need to associate to this network a proper execution semantic. So the good thing about these Petri nets is that those can be translated in different kind of systems, or we can use a different execution semantic. For instance, we can make ODEs, or we can execute in a stochastic fashion, for instance, associating stochastic rates to the transitions. What we choose, given the coarse-grained level of knowledge we started with, was the simplest one. So we choose to go for a Boolean representation of our network. And by doing so, we need to define what's the status, the state of network is at a given point in time. And we can represent that using a bit vector, in which each bit represents a gene in our network, and that bit being 1 means gene is expressed, or that bit being 0 represents that gene is at that point in time repressed. Now, we can represent these different states of the network, but the question is how we go from one state to the next. So here is a small example. We have, for instance, gene X, Y, and Z, and let's suppose to be in a state A in which gene X is actually expressed. So now we have two options. We can choose to execute T and move and basically express gene Y, or we can execute T prime and express gene Z. And as you can see here, that means going from state A to state B, in which actually Y is now expressed, or going from state A to state C, in which now Z is being expressed. We can encode this sort of execution in another graph. And this new graph, actually each node represents now a state in which the network is. And this is that graph, and that means that from A, by executing T, we can go in state B, or in A, we can execute T prime and go in C. Now, imagine that you can do that not only for this state, but for all the states, all possible states of your network. So basically, in our case, that means that if we have 11 transcription factors, and each transcription factor can be in 2 states, then it will be 2 to the power of 11, so that's something like 2048 states. So if you do that, and you do for all these states, what you will end up with will be the state space of your model. So all possible execution, all possible traces of your model. And let's suppose that that's our graph, which obviously isn't, because that's much simpler. But let's suppose that we end up with something like that. Now the important question is, can we actually map biological properties on this state space and check them? So we try to do that. And how to do that? We start focusing on something called strongly connected components. Those are subgraphs in which each node of the subgraph is reachable from any other node of the subgraph. That means that, for instance, from A, you can reach B, and you can reach A from B. So not only we were interested in the strongly connected components, but in the terminally strongly connected components. Those are the ones, are particular subsets of the strongly connected component, in which once you reach one of its states, it's not possible to reach any other strongly connected components afterwards. So basically, those are the attractors of your system. As soon as you reach them, the execution gets trapped there. And for us, that's the operational definition of a cell type. How is that? So the idea is that if you start from the real thing, so if you start from your cell, and then you measure your gene expression profile of that bunch of cell, you obtain something like this. Then the idea is you apply a threshold to binarize the data, and by doing so, you obtain now a bit vector. And we know that all our states in our network, in our state space, are bit vectors. So now we look where this particular bit vector, which corresponded to the real thing, is in our state space. And if it happens to be in its terminally strong connected components, then what we can say is that our model is able to recapitulate that particular cell type. So we did that for our model, and what we were able to recapitulate using this 11 transcription factor and this 39 interaction were just these two cell types, so the hematopoietic stem cell and the erythroblast. But then there was another detail that we found out. So as you've seen before, it might be that your attractors, your terminal strong genetic component, is not just formed by one state, that one that corresponds actually to the gene expression profile of your cell type, but might be composed by many others. For instance, in our case, the hematopoietic stem cell ones were composed by 32 substates. And so if just one corresponds to a particular cell type, you can start asking yourself, what are the other 31? And of course, those could be artifacts of your model. But we tried to explore whether there was some biological meaning on them. And what we found out is that if you look at the gene expression profile of single cell, instead of looking at a bunch of hematopoietic stem cells, what you find out is that the expression profiles of single cell can vary quite a lot. So our idea is, what if our model is actually to recapitulate the heterogeneity in gene expression profile of single cells? So what we tried to do then was try to correlate the frequency of expression in the measurements with the frequency of expression in the model. So intuitively, what we tried to do was to check whether genes that are basically never expressed in the single cell measurements, like for instance GATA1, which is never expressed, is always 0 in our model. Or if genes that are frequently expressed in the measurement, like SCLs, are also frequently 1 in the substates of our attractor, obtaining quite a nice correlation. So that was the first result. The second result was that we decided, okay, let's not limit ourselves to the attractors, to the analysis of the attractors, but let's see if by looking at the whole state space, since we already generated it, we can actually derive something more about our system. So actually, the question was, could we map the traces on the state space on the developmental tree? And of course, it's not possible to go, for instance, from the HSC attractor to the erythrocyte, just because, as I said, those are two attractors. So the execution gets trapped there, and there is just no path between them. And then what we tried to do was to try then, what if we try to simulate an inductive signal? So what if we take the HSC state, so basically, what if we take just the gene expression profile, the bit vector that corresponds to the HSC, and we try to simulate an inductive signal that flips 1, 2, 3, or 4 of these bits. So if we change them, simulating that there is an external factor that change them and trigger the differentiation, could we then be able to find a path in our state space that lead from there to a fully mature cell type? So we try to do that. And for instance, we can see that if we flip just 1 bit here, then we are able to go to the granulocyte. And if we flip 2 bits, then we are able to go to the erythrocyte. But that turned out to be a problem, because what the experimentally evidence suggests is that just by the expression of GATA1, so just by flipping 1 bit, it's then possible to force the erythroid lineage commitment. So it's possible to go to the erythrocyte. But that's not what our model was telling us. So then, what we decided to do was, well, what we can do with that. So there are 2 main explanations. So the first explanation was that we are just missing an important factor. So there is something else which is not in our network, which actually help driving the differentiation. Or in the other case, there might be that there is just, GATA1 is just missing a link. So it's not powerful enough to force the commitment. Of course, exploring the first option was quite complex because it could have been everything, basically. So we decided to stick with the second option and see whether it was possible to derive an hypothesis for a missing link. So a link that could increase the power of GATA1. And so we started looking again at our network and at our data. And what we found out is that by looking at the regulatory regions of FLY1 and GATA2, we could see that these 2 regulatory regions were quite similar. And since GATA1 is actually, we knew already, being able to repress GATA2, we just formulated the hypothesis, what if then GATA1 would be able also to repress FLY1, since these 2 regulatory regions are very close to each other. So we said, fine, we can just try that in silico. That's just a matter of putting a new edge here. and put the inhibition. So we did that, and then we tried again to compute our reachability. And now finally, it was enough just to express GATA1 to go from the hematopoietic stem cell to the erythroid lineage. So our model was predicting that that could have been really a possibility. And what we did was then going back to the lab and try to validate this hypothesis. So that was validated on a cell line in which we used to construct first a fly1 reporter construct with a control, or a fly1 reporter construct with GATA1 gain of function. So what we wanted to test it there was where if we transfected the cell lines with the reporter with the gain of function, we could see a significant inhibition of fly1, which is actually what is plotted here. So when transfected with GATA1, then we had a significant repression, pointing out that that actually is a valid link in our network. So, concluding, I think that what I wanted to show here are mainly 3 points. One is that, first of all, when you want to transform your biological knowledge into a computational system, you need a model which is intuitive and allows you to have a dialogue with your partner, your biological partner, and be able to encode all their experience in your model. And Petri nets, in this case, were quite helpful in that, as I show with the whiteboard. They could easily understand the methodology, and the methodology itself allows us to go directly to the modeling. Secondly, we built a network which was able then to recapitulate not the whole process. Also, we think that just part of the knowledge which is necessary to do that, but at least was able to recapitulate some aspects of the differentiation. But those were sufficient then to look at the network and generate new hypotheses which have been tested in vivo. So that brings me to the acknowledgements, and I'd like to acknowledge the MFN project, which provided the funding to enable computer scientists and computational biologists to get in touch with wet labs. And my group at the Freie Universität, the group of Yannick Scenarios in Switzerland, and the group of Bertolt Gottgens at the Institute for Medical Research. And of course, ICSB for providing Charles Fellow.
Hi. So I like this approach you're doing. So conceptually, it's very elegant. Just as a concern from practical point of view, so the possible combination of the rules from all these regulators to these genes presumably could be a large number. So how do you choose those to make sure that you're not overfitting?
So what I did there, we choose to just choose the experimentally validated one. So those are all the ones that are being experimentally validated. And when this work was carried out, those are the ones that we knew. Right now, we have a full, complete network. So basically, ChIP-seq and in vivo validation is being done for all possible combination of transcription factor. So we have now a complete network. This is a partial subset of that complete network. But we choose to restrict ourselves to the experimentally validated one, because otherwise, we would not be so confident anymore of what we are basically generating.
So just a clarification, so when you say this experimentally validated, you're not using the data, your single cell data or whatever, I mean later on to derive these combinatorial rules, right?
So that's an interesting point, because we are also working on trying to automatically generate this network out of the data. This is an interesting aspect for which we have a project running on this. But in this particular case, it was just about, let's say, mining the knowledge of the biologists, which of course were intimate with this sort of data. And because of that, then we needed a language which would allow us to establish this sort of communication.
Thank you.
Thank you. Pardon me, excellent paper. I was just wondering, in your biological validation, if you have just presented a partial part of it, because I would have expected perhaps that you would show the reverse with siRNA or other manipulations. And currently the conclusion is not that you have a direct inhibition between these two partners, but direct or indirect throughout the network.
No, you're right. So I think more validation should be done to assess that that's exactly the link in these two, that basically there is a direct link between these two, because from what I show here, there is no experimental evidence that shows that. So more has been done. But
sure. Have you tried to also look into the robustness of your prediction by introducing at random certain links?
That's, I think, a very interesting point, a very interesting question, because we tried that. Because also at some point, we had the question, what if we can generate all the missing links, right? What if you can actually reproduce the whole differentiation tree? What we should do to do that? So we tried doing that. And I must say, it's much harder than it thinks. So as soon as you introduce a new link, you are much more likely to disrupt the whole network than to keep things the same. That's also why, just by introducing this link, we were quite surprised just to maintain the result, because that's an important part. We didn't disrupt the result by just introducing the link. We just increased the agreement of the results with the biological evidence. But it's very likely that when you introduce a link, you just disrupt something.
Okay, thank you.
Okay, thanks for an interesting talk. How much tuning and background knowledge did you use for the thresholding, and how sensitive is the network for that?
That's also a very important part. So I must say that in this case, we just had few data across the whole setup, because at that point in time, for instance, single-cell microarrays were not that popular. Right now, the partner at Cambridge just published a paper with about, I think, 800 single-cell gene expression profiles. So at this point, we would have much more knowledge to assess these sort of parameters. At that point in time, we again just, as you can see also from the previous one, we just use a sort of binary discretization without being able to really adapt it because just of the scarcity of the data. So we just ask our biological partner to assess the threshold, confiding that they have the expertise to do that. But of course, the best way would be to have the enough data to then assess the threshold, and maybe now.
So the work that you showed had this transformation from the Petri net to the state space. And I'm just curious, if somebody were to try to apply these techniques in some other system where they have, say, more than 11 transcription factors mapped, you can imagine at some point it won't be possible to explicitly represent the entire state space. And I'm curious if there's any way to apply some techniques from symbolic model checking to answer the same types of questions that you got at with an explicit representation of the entire state space.
Yeah, no, I think that's a very interesting, it's a very interesting question. I think one of the points that we try to make with this work is that most of the time when people work on this sort of Boolean model, they focus on the attractor themselves and not too much, let's say, on the wall connection between the attractors. So I think that's an important point. And of course, at some point when this model will increase, you will not be able to treat the state space by itself. So we'll have to use some symbolic technique to do so. We personally didn't investigate that line because we tried more to tackle the biological questions, let's say. But of course, some improvement or some technique from the symbolic model checking world, let's say, would be very interesting to be applied to this sort of mechanisms.
Okay.